Dr. Weng holds the Li Weibo Chair in Biomedical Research at the University of Massachusetts Chan Medical School, where she is the inaugural chair of the Department of Genomics and Computational Biology. She received her PhD at Boston University, working in the lab of Charles DeLisi. In 2008, Dr. Weng joined the UMass Chan Medical School, where she established a new program in bioinformatics and integrative biology. She was elected a fellow of the American Institute for Medical and Biological Engineering in 2013 and of the International Society for Computational Biology in 2020. She also co-founded the biomedical startup Rgenta. As part of the ENCODE and psychENCODE consortia, our lab has developed important computational tools for integrating multi-omics data to understand the molecular mechanism of gene regulation. Please welcome Dr. Weng.
Thank you so much for inviting me. It's such a pleasure to be here. I have to say that I am a beneficiary of Gary as an outsider, and I first met Gary during a conference, I think in 2000, and I was just starting my research lab as an assistant professor, and I read this review paper by Gary, and then I went to a meeting and I saw Gary at a distance. I said, okay, maybe I should approach him and talk to him. And so I was kind of shy. Much to my relief, Gary was so nice and so kind and explained to me. So I was like, I was at the beginning of doing genomics research. My PhD was more like in biophysics. So at that time, I was getting into DNA sequences, and I was like, how do you make a position weight matrix? Do you have to do something special when you have not many sequences? What do you do? And then Gary very patiently explained to me that you have to threshold, take the highest affinity binding sites. And then that makes a big difference. If you don't get the highest affinity binding sites, you're just going to be garbage in, garbage out. So it piqued my interest. So I switched into genomics at that time. And every so often when I go to a meeting, if I see Gary, I'll try to make some conversation with him and get another dose of an insight and encouragement. And I remember talking to Gary about this last paper here, Takas was the first author of that paper, and Martha was on it too. And I was like, oh, am I not supposed to use a position weight matrix anymore? And he explained to me again, oh, they are actually quite good. Unless you're looking at specific interactions, you still continue to use them for your work, it will be fine. After listening to all the talks today, apparently I didn't go to any of the fun meetings. I only went to the scientific meetings. Nonetheless, I'm grateful for all the benefits I got from Gary. Yesterday I was searching for any paper I could be on, and I was very lucky to find this paper with my colleague Scott. And at least I can claim I have co-published with Gary. So today I'm going to continue to talk about research that I took on since I met Gary. So I'm very interested in the same general research area of gene regulation. So obviously we want to go from regulatory elements to gene expression. And also we are very motivated by finding all these genetic variants, difference in DNA sequences in our genomes. How are they affecting our traits and disease propensities? So I did that by joining a very large research consortium, ENCODE. You have heard about it. It was a 2-decade collaborative action, and I was lucky enough to be involved in it as a principal investigator for both the entire time, and then for the second decade, I was leading the data analysis center. So as such a role, and we had these unenviable task of heading the consortium to put together a consortium paper. So 3 students of mine were key analysts of the project. Among the many other analysts, and we published work that was just mentioned by Steve. So I was happy to hear that it's useful and candidates regulatory elements. And it's a very awkward acronym and required by the reviewer. So we had to add the small c in it. So C stands for candidate, and we still haven't, we can't escape the small c will be stuck. Hopefully people don't mind at this point. So we used a boatload of ENCODE data, thousands of ChIP-seq ENCODE data, and of transcription factors and histone modifications and DNA-seq experiments. And altogether, I was going to brag about how clean the DNA-seq experiments in ENCODE are until I hear Steve's data, and then now I'm not going to brag about it anymore. And cut and seq, cut attack are apparently better. It looks way better on the screen. So we took DNA-seq data. We took these regions, and you can see they line up really cleanly. And then if you take thousands of such data sets, you can get regions in the genome that are representative hypersensitive sites. And then we add, we have millions of them. We have more in human than in mouse, just because we have more data sets in human. They should be roughly the same, millions of them, like 2-point-some millions. And then we add on top of them, you see DNAs on the top. We can add key histone modifications such as H3K4 trimethyl, H3K27 acetyl, and then the CTCF is the insulator binding protein. And altogether, we can define these CCREs. Right now, at the current version, we have 2.3 million in humans and 0.9 million in mice. So the whole point of these CCREs, it's like it is a repertoire of candidates, regulatory regions in the genome. And you can see in the boxes up there, CCRE row. And I don't know how to point. Does this point work? Okay, never mind. So in a particular cell type, out of the 2-point-some million of them, only like 150,000 of them could be doing things for that specific cell type. As an example here is a hepatocyte. All the colored ones are the ones that are likely to be functional in that cell type, and the gray ones are just not used. So the idea is you can pick and choose between different cell types, the different subsets of the 2-point-some million of these CCREs for a cell type specific regulation. So here for hepatocytes, I show transcription factor HNF4A is the gene that's important. You see the locus is entirely lit up. However, if you go to a neuron, this locus is quiescent, even though the insulator region still are insulating there on the side. So I want to also include other work that's related to what I have learned from Gary. So one thing is the evolution. So I'm going to, okay, so let's explain this a little bit. So we take these CCREs, and then these are different subsets of CCRE, the promoter, enhancer, whatnot, and different colors over there. Never mind the acronym. Again, consortium product, hopefully useful too. And so at that time, we had 100 vertebrate genomes, and you can average them. They are more conserved than the gray line at the bottom. They are conserved. However, if you look at between human and mouse, because we have CCREs in both human and mouse, only 1/3 of them are conserved between human and mouse. So we were wondering, like these are these low philo P CCREs truly unconserved, or would they show conservation with more genomes in more recent evolutionary time? So if you're paying attention, I want to rebut what Bill said earlier. Even though it's all good, Gary's aligning all DNA sequences, but all the information is in the proteins. I'm going to rebut that. And also, I'm agreeing with you. However, it depends on where you're looking. Okay, so if you're looking at the distance of vertebrates, you're not going to find DNA conservation signal. However, if you look at the sequences in mammals, we actually look at 100 million years of evolution, as you mentioned, we can see DNA conservation. So the dashed lines are the lines you saw earlier. for 100 vertebrates. If we go to 240 mammals over there, and all the lines get higher in terms of philo P score. So we have a paper in this Zoonomia package last year looking at the conservation and evolution of these CCREs and other transcription factor binding sites throughout mammalian evolution. So we made the CCREs into these triangle by design, with the y-axis indicating how strongly they are aligned across the 240 mammals, and the x-axis how poorly they are aligned across the same 240 mammals. So by design is simplex, and then the G1 group at the corner is the most conserved, and G3 group at the tip is human-specific, but primarily primate-specific. So if you compare with the random regions, you see G1 is a lot redder for CCREs, and G3 is much lighter than random. You see this has human-specific. It's much more in random regions. So we are in a big data now, so we can take 1 million CCREs. That was version 3 in the paper, in the Nature paper, and then 1 million versus 240, and make a big matrix and feed it to algorithms like UMAP, which is reducing the dimension. And you can see there is an entire spectrum of evolution of these CCREs from the most conserved tip to, which is group 1, most conserved across all the mammals, to the ones that are blue, which are primate specific. And the surprising thing to us was basically this looked to me like a shrimp, which had been blown up at the head. And the blue part, the primate specific parts, form discrete clusters. So what are they? And we can do the same for transcription factor binding sites. So we actually did that for each of the TF binding sites for nearly almost 400 transcription factors. This is HNF4A. By now, you probably have realized this is one of my favorite transcription factors. And you can do this for that binding sites. So the HNF4A binding sites are smaller, as you probably have realized now, like 8 base pairs long. And then CCREs are 1 nucleosome size. Okay, just in comparison. So all these clusters, discrete clusters, correspond to gains and losses of the binding sites throughout the mammalian genome evolution. And this is the tip of the 240 mammalian evolutionary tree. And each cluster corresponds to different ways of gaining the site. Like the cluster A is for gray apes, cluster 2B is for gray apes and old world monkeys, for example, and so on and so forth. And why are they like that? It's because they are brought about during evolution by different transposable elements. And so tribute to Ting's work and transposable elements. And different clusters, they're basically radiating across the genomes because of these mobile elements. And if you look at all the group 3 are the primate-specific sites, 86% of them are derived from transposable elements. And you see the blue color ones primarily from line 1, LU, and LTR elements. So, but those ones are the not conserved ones, they are primate-specific. And so if you look at the other extreme of the conservation, the evolution, if you look at the conserved sites, we can detect them for each transcription factor binding sites. These are the sites within the ChIP-seq peaks of the transcription factor, and they form this bimodal distribution using the 240-way philo P score. And they look different between transcription factors because we think the evolutionary speed is different for the sites for each transcription factor. So for each one, we fit a 2-component Gaussian model, and we can get the sites that are constrained. We call them constrained. And they are at the tip of the most constrained sites of the UMAP. And then you can do all kinds of analysis on the constrained sites. We can show that they are most likely to be functional. They have position weight matrix that's more conserved across the genome than the unconstrained sites, even though these analyses are human-centric. So all these sites, when I say constrained, means these sites exist in the human genome, but they are not constrained across the mammalian genomes. And if you use the ChIP-seq data in the respective mammals, you can see that the constrained sites are the sites above the gray line. Those are actually bound in the respective species. These are HNF4A again in liver cells. All of them are in liver cells. And we did other analysis and so on. We moved on and we wanted to combine the CCREs and single cell data to study the human brain. We took, as the introduction said, I'm part of the psychENCODE consortium as well. So we have quite a bunch of brain-specific data, single cell, bulk, Newen, FACS-sorted data. So we took the CCREs and then we added a B to it, B-CCREs. A subset of them are likely to be functional in the brain. We have adult data, fetal data, and then Newen plus. Newen is a nucleus membrane. It's actually an RNA binding protein on the nucleus of membrane and signifies neurons versus non-neuronal cells, glia. So Newen plus are neurons and Newen minus are glia. So we can classify these using psychENCODE data. And so these B CCREs, if we look at them, we have adult data, fetal data, and shared data. And then we have CCREs that are not brain specific and then non-CCREs. And you can see that the fetal specific CCREs are most likely to be validated by the Panaccio et al. Transgenic mouse assays. And those are fetal and in the brain. And then we can use single cell attack-seq data to show which brain-specific CCREs are likely to be cell type specific using the different types of brain cells, astrocytes, excitatory and inhibitory neurons, microglia, oligodendrocyte precursor cells, and oligodendrocytes. And then we can use GWAS data to show whether or not they are enriched in different brain-related or non-brain-related genome-wide association traits. And you can see that the brain-specific CCREs are most enriched in brain-related GWAS traits, and only microglia, which is the brain-resident macrophage, the brain-specific CCREs active in those are enriched in both brain and non-brain traits, with non-brain traits specifically being immune-related. And we can use, now it's AI, right? We're all going into AI now. So we're using variational autoencoders, and all these brain-specific CCREs, if you train a VAE, variational autoencoder, and it can distinguish all these subsets of the brain-specific CCREs just purely based on their sequence information, which is pretty remarkable to me. And we have collaborated with Anshul Kandaji's lab using their COMBPNet, which is a convolutional neural network model. And you can see that this is one CCRE. And then if you train a model using the genome-wide data and then go back and decode it, and you see the residues that are important for the training and testing, this is completely unsupervised. And you see the residues remind you of transcription factor binding sites. And I want to mention that from Martha's talk about multiple weak sites near a strong binding site, it's quite remarkable. You see a whole train of CRAB1 binding sites in these CCRE, the repeat, like 5 copies of them. We see that a lot, especially in open chromatin regions. So we can combine the single cell attack-seq data with the single cell RNA-seq data so that we can deconvolve the transcription factor paralogs that's functional in each specific cell type. For example, in excitatory neurons, you can have neural D. You have a lot of neural paralogs having enrichment for motifs, but the paralogs all have the same motif. But if you then combine the single cell RNA-seq, then you can pick out which paralog is the one that's likely to be functional. So just very quickly, you can go back to the cell types. You can build a convolutional neural network model for each of the cell type as indicated by the lines. So now we have six models. Each one is cell type specific. And then you can go back and score the sites. So for excitatory neurons, only neural D only stands out for excitatory neurons. And for microglia, you see the purple color, it's only the SPI1, which encodes for PU.1 is the protein, only stands out. So the key thing is it's actually quite interesting. These binding sites reflect the lineage of these cell types. So if you see a high score for, say, oligodendrocytes, like the other glial cells also have not the highest, but also high. You see astrocytes, OPCs. Okay, so if you see neurons, you see inhibitory neurons, excitatory neurons is the next to follow on. And except for microglia, everything else is just flat because microglia is its own lineage. So that is my last slide, and I want to go back. Oh, sorry. I have a couple slides saying making the tools available. So we have a tool called the Screen, and I'm just going to skip over. And we have a tool called Factorbook. So if you are interested in our work, check those out. Those basically my work. So at the end, I want to say I'm very grateful to be honored, grateful and honored to be part of the celebration of Gary's scientific legacy. From more or less an outsider and someone who has benefited from Gary's generosity and insight, I feel that he's a legend. He has pioneered the field and has created so many tools that we are still using today. The tools are as good as the new ones, and I'm still making PWM and in my publications. And he has mentored so many students and scientists near and far. And I remember this one benefit I took from Gary when I talked to him over time. He says, oh, you have to keep on thinking ahead because the field is going to change and there are all these new technologies. Don't be too fixated. PWM is good, but think about new things. So I have taken from him that and has benefited me tremendously. Thank you.
We can take a couple of questions before lunch.
Yep, Zhiping, that was great. Your talk made me think about the numbers for the total amount of conservation in mammalian genomes. People think that maybe 5 or 10% of the genome is conserved, 1% of it is protein coding, the rest, we've waved our hands and said, oh, it's probably regulatory regions. You've got a couple million CCREs times maybe 100 or 200 nucleotides. Seems like that's the explanation. Do the CCREs account for the conservation patterns we see in mammalian genomes?
Yeah, if you take our CCREs, probably amount to like 20% of the genome. It's kind of big, but not all the positions are conserved. So inside there are bits and pieces that are conserved. So I mean, I think it's more than 5 to 10%, but depends on where you look, whether or not you look within mammalian, you know, it's a window, right? Like all the blossom 62, why 62, right? This is evolutionary distance. Depends on, you know, like your hidden Markov models too. You're trying to evaluate the distance. Depends on what you're looking for. You should look at the correct distance. So I would say definitely we are trying to hone in to the conserved sites, but there are like 20%, right, of the sites we see are primate inventions brought about by transposons. They're not fixed in our genomes yet. Some of them are functional. They are bound. They are like, these are all in ChIP-6 peaks. They are not computational predictions.
But it's not looking to you like there's another class of sequence conservation that hasn't been explained yet. The CCREs look like they're the main explanation for the conservation. Is that right?
I would like to say that, but there might be people who, yeah, who have other non-coding RNA and, you know, other species of biological moieties that's important to be conserved.
Any other questions?
Yeah, thank you for the great resource you provided. So I'm wondering, like, how big because your CCRE is small, like you said, 20%. Like when you use a single cell attack-seq, do you enable you to find more like a regulatory sequence? And how big is the regulatory space in the genome?
I think our collection of CCRE is pretty comprehensive. If you take the single cell attack-seq data, our coverage is like 80 to 90% of the single cell attack peaks. And the 10 to 20% that are not covered by us, you can look at the attack signal. They tend to be on the low end of the attack signal. They tend to be not as open. So I think that's—
How big do you think the regulatory space is? Someone estimate like you get 20% coverage.
20%.
How like some people saying 80% of the genome? Like what's your, how
20%.
many do we need to sample more?
Yeah, I mean, you might be referring to the ENCODE phase 2 publication, about 80%. A lot of the 80% comes from transcription, and then a lot of it is intron. Obviously, introns are transcribed. So if you look at that biochemical signature of transcription, you get to 80%. But how much of the intron is functional, truly functional in a biochemical or in biological sense? I mean, it's up for debate. They're transcribed.
Thanks.